All right. So, excuse me, I ended the last video talking about how the lac operon was kept in the off position. All right. Remember what we have? We have our promoter region, we have our operator region, we have our genes. And let me label these again. Promoter, operator, these are our genes. All right. That repressor protein is made, and because it's floating there in the cytoplasm, it can sit down on the DNA associated with the lac operon, sits on the operator, and essentially acts as a physical block so that we don't get transcription of our 3 genes needed to break down lactose. All right. Main difference between off and on. On, we've got lactose present now. We've got lactose that's in the cytoplasm, all right? That represents essentially an energy source that the bacteria wants to use. So now it wants to turn on those genes because it wants to take advantage of that lactose being present, all right? Now in terms of the on position, our repressor protein is still going to be made, all right? But what happens when there's lactose present in the environment? Lactose can bind to our repressor. And if you notice now, I've kind of changed the shape on the repressor. What happens when lactose binds to that repressor protein? It results in a conformational change in the protein. It changes shape, all right? That repressor now doesn't have our little bits that can sit down on the operator region in our lac operon. What that means, because we've got our promoter here, this is still our operator, R is our repressor, all right? What that means for the bacterial cell. When RNA polymerase comes along now, there's not that physical block there. So what ends up happening, that RNA polymerase chugs on down and it starts making the RNA transcript for those 3 genes. So we've got Z, we've got Y, we've got A here, all right? Once we've got the RNA transcript, our ribosome can come along and make our proteins, all right? So now we've got the 3 proteins made that are going to work on breaking down lactose, all right? The beauty of this system, when lactose levels get low, there's no longer lactose available to bind to the repressor protein. That repressor protein will be made, now can sit back down on the operator, can shut off transcription of those genes, all right? So this is what it means for the lac operon to be an inducible system. It's induced to undergo transcription and translation by the presence of a molecule. In this case, the example of the molecule is lactose. Lactose binds to the repressor. Repressor cannot bind to the operator. That allows RNA polymerase to come in, chug along, make the RNA transcript. RNA transcript is translated into proteins. That bacterial cell is now able to break down that disaccharide as an energy source. All right. Hopefully that makes sense. If it doesn't, let me know. I've got another type of operon that I'll have a brief video on, the trip operon. It's known as a repressible operon, and I'll talk a little bit about the difference between something that's inducible versus repressible. Lac operon, This is our inducible system. All right? And the inducer in this case is the lactose. All right? Thank you for your attention. Let me know if you have questions.